Okay, let's talk. So we all have microbiomes. We have body sites that are not sterile. We also have body sites that are sterile. Yes. This is week 9. So if you look at this table, and I have no intention of reading it to you, I think you can read yourself perfectly fine. You can see that there are some question marks. So these question marks show the sort of disagreement between different scientists, whether or not we can call those sites sterile. For instance, there were traces of bacterial and viral RNA were found in the brain. Traces of bacterial and viral DNA were found in the heart, and lower respiratory tract, which is your bronchioles and lungs, is known to have microorganisms coming in with the air that you inhale. So when we use the term sterile, we refer to the fact that all those body sites summarized in the table do not have a resident microbiome. So what is the resident microbiome? The resident microorganisms normally inhabit different body sites. So what are those body sites? As I often say, if you take 2 microbiome researchers and ask them a question, you're going to get 3 answers. Okay? So we definitely have a microbiome in the mouth, in the nose and upper respiratory tract, which are actually quite different. Eyes, well, I consider it a part of the skin. Gut, for sure. Vaginal, sure. Now, the only one that concerns me here is placenta, because placenta is supposed to be sterile. Okay? Good point. Placenta itself is probably sterile, but there is a transmission of microorganisms from mother to child. Okay? Now, microorganisms that can be found in humans, let's say this way, can be resident, as I just mentioned. We acquire them from different sources. We start at birth, and then we continue with environmental factors and food and water and air, whatever. Now, here's the question that I have for you, folks. Let's say you have 2 newborns. One was delivered through the birth canal, and another one was delivered by C-section. Question number 1, are they going to have different microbiomes? And the kid that was delivered through C-section, where did he get his first microorganisms from? The doctor. The doctor. And the difference in microbiomes persists until about 3 to 5 years of age. Then it basically equalizes, because the experiences are pretty much the same. Okay? Now, there are microorganisms that can transiently colonize you. Transiently meaning that they will become a part of your microbiome for some time, and then they will disappear. Okay? Well, we call them transient, right? So transient microorganisms, as you can imagine, can be pathogenic, causing some problems. And they may not be, just hanging out, you know. As a matter of fact, if you go to another country, there's a pretty good, and try different foods and interact with different people. By different people, I mean people not from your country, the locals. Your microbiome will change. And theirs will change too. Okay? Now, microorganisms are categorized into two big groups based on their ability to cause the disease. One is the primary pathogen. It can cause a disease in an average healthy person. And then there are opportunistic pathogens that will give troubles to people with impaired immunity. Okay? So, if you think about like various microorganisms that exist in our gut, for example, I don't remember if we worked with this, Enterobacter genus, or that government conspiracy that I told you about Serratia marcescens. Serratia marcescens is a classic opportunistic pathogen. For the most part, it doesn't affect people with normal immune response, but when this immune response is suppressed, either by another infection or immunotherapy, then those pathogens become dangerous. Okay? Now, before we move on to very interesting conversations about microbiome, if I would ask you before I made the statement that sterile sites are more susceptible to infection, I would ask you, what is more likely? Like, which organ is more susceptible to infection, skin or, say, liver? What would you tell me? Well, it's not that easy, because, well, yes. So skin, although it is exposed to all the elements, I agree with you. Due to the competition from your normal microbiome on the skin, does that make sense? Any incoming pathogen would face serious issues with overcoming that competition. If that same pathogen somehow makes its way to the skin, you're boned. There's no competition. Ideal environment for microorganism to grow. So sterile body sites are actually more susceptible because they don't have that microbial competition. So where are we going to find microorganisms? Well, skin and eye, upper, and I won't emphasize upper but not lower respiratory tract, because lower respiratory tract does not have resident organisms that stay there forever. Oral cavity, obviously. Stomach, not particularly well inhabited. Intestines, yes. Vagina, and distal urethra in females and males. Okay? Does that make sense to you? So, another question that I want to pose for you. You're familiar with digestive tract, right? So, there is a fair amount of microorganisms in your mouth and in your throat, but not as much in the esophagus. And then again, fair amount in the, well, there's one species, Helicobacter pylori. It can be in your stomach, and then there's a ton of microorganisms in the intestines. Can you propose any idea why esophagus and stomach aren't terribly populated? So stomach acid, yes, acid is pretty bad, yes. And believe me, Helicobacter does not like acid. It doesn't live in the acid, it lives in the epithelium. What about esophagus? If things pass through esophagus, how is it different from intestines? Yes. What was your idea, Amanda? It all has mucus layer, but you're right, Gemma, that you're right that the fast passing and its stratified squamous epithelium so it gets peeled off all the time. So microbes have really tough time to stick around, you know. And one may argue, well, there's a stratified squamous epithelium in the mouth and in the throat, but the passage goes A, much slower, B, you have constant inhabitation of those sites from your nose because the snot comes down if you have a bit of a runny nose. Okay? Now, a lot of diseases are associated with alterations to microbiome. For instance, here, I'm showing you the changes in oral microbiome associated with various dental diseases. Okay, strep mutans, dental caries, enterococci associated with endodontic disease, Fusobacterium, oh, that's my favorite, gingivitis, okay, and periodontitis, Porphyromonas, okay. So why pay so much attention to this? Anyone in this room is planning to join dental hygiene program? Yeah. So this microorganism here, Porphyromonas gingivalis, was tested in mice for its ability to somehow trigger Alzheimer's disease in mice. Short story, it does. If you infect mouse oral cavity with Porphyromonas gingivalis, they will develop gingivitis, and they will also demonstrate pathological changes in the brain that are basically identical to the ones in the human brain. So next time, or when you are in the program, here's the point for education. Okay? Make sense? Streptococcus mutans is associated heavily, this guy, heavily associated with increased cardiovascular disease in humans. So, to summarize, yes?
What about that periodontitis has some effect on the human, like the mice?
It's hard to study it in humans, you know? You can, like, I would love to give you a question. What kind of study would you propose? And you can pretty much, if you have good electronic health records, then you can try to see if there's any correlation with the diagnosis of gingivitis or a periodontal disease and the early or earlier onset of Alzheimer's. Make sense? Yes. This is PDF. So if you're looking at PowerPoint, I think PowerPoint misses one or two slides. I'll update then. Okay, I will update the PDF. Okay, I may be a bit of a mix-up. Okay, so to summarize, what you need to know about microbiomes. First, microbial compositions at different sites are very similar among people. Now, different sites on the same person have different microbiomes. I'm going to put it here. Sorry. So same sites are similar in different people. So what does that mean? Your skin microbiome is different from your oral, from your vaginal, and so on and so forth. However, if you test all of us here for the general composition of our gut microbiome, it's going to be very similar. Okay? Now, microbial alterations are often associated with the disease. Okay? Again, this is a really cool table. You can read it. I'm not going to just read it for you. Does that make sense? And one more thing in addition to those 3 points is that human microbiome heavily influences human metabolic, immunological, and other developments. And I have some data to support that claim. So first of all, if you look at the composition of the skin microbiome, which is this image on the right, it shows you that it varies even on the same person depending on which area of the skin we're talking about, which is, to me, fascinating. Okay? So basically, think about this. The butt and the crease under the butt have two different microbiomes, like different composition. It's kind of crazy. Now, how is this microbiome associated with the disease? So I'm going to comment on the data. We're not going to totally, explain what's going on. So what you see in this panel and these panels is the association between microorganisms found in the skin in people who have atopic dermatitis. Okay? Sounds good? So this, the very first one, this graph, shows you the diversity of the populations at the baseline during the flare of the dermatitis and after the flare. Okay? So if you look at these dots, right? The blue, the yellow dots. The greater the diversity index, the better. So it's pretty diverse. Post-flare, it's also pretty diverse. But during the flare, the diversity decreases. Why? So these are different groups of individuals. Controls are people without atopic dermatitis. Baseline are the people with atopic dermatitis when they don't have a flare. Now, look what happens when they have a flare. Do you see the purple color absolutely dominating? That's the phyllococcus. They take over the skin. And then, gets better. And which staphylococcus exactly? So, this middle panel, that one on the bottom, can you see it? It's almost entirely I don't know what you're going to call this color, like, magenta, right? Staphylococcus aureus. Okay? So, now, can we say that Staph aureus causes atopic dermatitis based on this data? Can we establish a causative association? No. We can only say that the flares of atopic dermatitis are associated with the higher populations of Staph aureus, and we can say that when we treat, it definitely reduces proportion of Staph aureus. So maybe Staphylococcus aureus contributes to the development of atopic dermatitis. Okay? Now let's get to the gut. Honestly, studying microbiome of the gut is a bit challenging because we only know 1,500 species. How many species of bacteria that are living there are unknown? No idea. Okay? What we do know, first of all, is that the microbiome of the gut shapes the immune responses. There are mice that have no gut microbiome. They have impaired immunological development. Okay? Now, it's been shown that probiotic bacteria, such as Bifidobacterium and Lactobacilli, can be protective in patients with inflammatory bowel disease. So, a lot of studies established the link between the gut microbiome and various conditions. Now, please, I want you to understand. I'm not going to focus on those individual studies or reviews on the exam. I give you a background that explains the importance of microbiome in different sites, and I hope to instill in you a thought that we should consider microbiome whenever we look at a disease. Does that make sense? So, I'm trying to prove to you that microbiome is really important for immunological and metabolic development. Okay? And in terms of metabolic development, I love this paper. So, let's take a look at this bar graphs. It's really cool. So, these are mice that are placed on the conventional diet, CHO, and the Western diet. Would anyone venture a guess what is a jargon name for the Western diet? Company name. McDonald's diet. High fat, high cholesterol, high sugar. Okay? So, if you look at this data in mice, and it would be fair to say that mice normally do not consume enormous amounts of sugar or fat. They eat grains and grass and stuff like that, right? But we feed them anyway. So, the group of bacteria called bacteroidetes, gone. Is it good? Is it bad? We don't know, but there is a massive alteration of these bacterial populations. Okay? That makes sense? Now, if you place mice on the Western diet, the diversity also decreases, which is always bad. The less diverse the bacterial population is, the worse. It's just known. I'm going to give you some other off-the-cuff comments. And finally this, take a look. So mice that are placed on the regular chow, they increase their body fat to about, they become two less fatter than the mice placed on the Western diet. Which makes sense, right? If you eat someone with, food from McDonald's, they'll probably become fat. But that's not the end of the story. I mean, it's the end of the story on this image, but it's not the end of the story overall. It's been shown that if you transplant the microbiome, fecal microbiome, basically fecal transplant, from fat mice to normal weight mice that were from mice on the Western diet to mice on the regular chow, mice on the regular chow will become obese. And vice versa. If you transplant feces from the normal weight mice into obese mice start losing weight. Okay? I mean, I'm not suggesting but maybe it's something to consider, yes. We don't know. So the idea is not the appetite as much. The idea is that it's different energy processing in the gut. You may wonder if that was done in humans. It wasn't. Fecal transplantation for the purpose of losing weight wasn't done in humans, but there was a study when researchers managed to identify several identical twins, the pairs of identical twins around the US, and in each pair, one twin was normal weight and one twin was obese, and they compared fecal microbiome. Lo and behold, they saw this. In people with obesity, the bacteroides percentage is much lower. Does that make sense? Not the end of the story. Most people consume fairly carbohydrate-rich diets. Does that make sense? So they would have something like this. If otherwise they don't eat 5 pizzas every night. I'm talking about normal products that people eat, but usually it's fairly rich in carbohydrates. Does that make sense? Now, when people switch to high-fat, high-protein, low-carb diet, the percentage of bacteroides skyrockets, and percentage of firmicutes goes down. And what people do when they move to that diet, high-fat, high-protein, they lose weight. So what I'm trying to illustrate is that it's really hard to separate the effects of a diet itself, regular human physiology, changes in the intestinal microbiome and potential products that those bacteria release that may, as who asked about the appetite? Yes, and potentially chemicals that may act on hypothalamus and suppress appetite. There is an idea of the gut-brain axis in which microorganisms present in the gut somehow produce signals that affect the brain. Okay? Now, that may sound very implausible until you look at this. So I want to start with that data, because this data suck. By suck, I mean it's data, it's just not super solid. The advantage, though, this study was done in humans. So these authors looked at people who were on autism spectrum versus normotypical people. Okay, do you follow? And they found that in people who are normotypical, the percentage of those bacteroides is greater than in people on autism spectrum. By the way, bacteroides are the ones that are associated with low-carb, high-protein, high-fat diet. Am I saying that the typical American diet causes autism? I do not. Well, do you know one of my favorite correlations? If you will plot the consumption of ice cream versus deaths caused by drowning in pools, it's perfect. It's almost a straight line. You can look it up. It's a perfect correlation between consumption of ice cream and drowning in pools. You know why? Summer. This is when people eat ice cream and swim in pools. So what I'm trying to illustrate, correlation doesn't mean causation. But if we see the changes in the microbiome that are caused by a diet and similar changes observed in people who are on the spectrum versus normotypical folks, I personally think it's worth, maybe if we change the diet, it may alter the behaviors. I mean, it's possible. Much better than just blame Tylenol or circumcision. Circumcision doesn't work. I have a perfect yes, no. Because Europe does not circumcise. In Europe, they don't generally circumcise newborns. Look at their railroad system. In the US, we circumcise everyone, and our railroads suck. So it's definitely not circumcision. Now, this is one of the most elegant studies that I have seen that establishes the gut-brain link. So, these scientists acquired several mouse models of autism. And I'm very adamant to say it's not mouse autism. It's a model. They exhibit autistic behaviors. Okay? And models could be environmental, so they treat it with something, some of them genetic, some of them... Do you know what idiopathic means? I prefer we have no fucking idea. Like when somebody says, oh, it's like idiopathic pulmonary hypertension, for the last 50 years we didn't manage to figure out what causes it. Yeah, that's idiopathic. So mice that exhibit autistic behaviors. So they added Lactobacillus reuteri to their water. And that treatment, it's lactobacillus, it's a probiotic. That treatment alleviated the autistic behavior in different mouse models, which is cool. But they really wanted to know how bacteria in the gut can communicate with the brain. So, for ANP1 aficionados, which nerve connects the brain and the gut? They cut it. Guess what? They give those mice Lactobacillus reuteri, mice with a cut vagus nerve, no effect. So it's definitely the vagus. So next question that they asked, okay, which neurotransmitter? So they produced mice that lacked the receptor for the neurotransmitter called oxytocin. It's a hormone, same, yes, it can serve as a neurotransmitter. Lo and behold, oxytocin receptor deficient mice were not affected by the treatment, by the Lactobacillus reuteri. Honestly, I didn't follow through, but, and please, I'm not suggesting that, people should go and buy that stuff. But I wonder if people went and bought that stuff. Because Lactobacillus reuteri, you can buy it in the United States. It's pills, you can buy it online, you can buy it in the stores. Like, it's a probiotic, it's absolutely benign bacteria. Which is kind of cool. Yeah? Yeah.
This is all my sister needs.
That was, wow. But I appreciate the honesty. That was great. Okay, now let's jump to the concept of infection. We're going to talk about different aspects of it. Infection implies the replication of a pathogen, period. It doesn't imply that there are going to be symptoms or signs. Clear? Infection without symptoms or signs is called asymptomatic. Now, infectious agents, bacteria, viruses, fungi, whatever, can cause communicable or non-communicable diseases. I'm highlighting basically terms, that definitions of which you must know. Communicable disease is the one that can be transmitted from person to person. Or if we're talking about diseases of animals, from animal to animal. Does that make sense? Non-communicable disease obviously can't. I can give you some examples. You're familiar with peritonitis? Yeah, just as a condition. It's a non-communicable disease. If somebody, it's an infection, when appendix ruptures and the stuff spills out, it's an infection, but you can't give peritonitis to somebody else. Have you heard about legionellosis? Okay, Legionella, you will, in the summer. There will be cases at Cedar Point. Yeah, every year. Yeah, every year there are going to be cases of Legionella at Cedar Point, the water park. Yeah, because of the stagnant water. Non-communicable. It's a pneumonia, but it's not transmitted to other people. Okay? That makes sense? Now, you may remember brain-eating amoeba, Naegleria fowleri, non-communicable. Okay? Now, another term that we use for infection, contagion, so contagious. It's not a strict scientific term, but it is used in sentences such as this microorganism is more contagious than another microorganism. Easier to transmit, infects more people. Okay, so now can we measure it? The brief answer is no. Well, indirectly, yes. So there's a concept, not for the exam, of the secondary attack rate. So let me explain. So basically, imagine we have two families. Same size, more or less similar families, right? And a person in a family gets sick. Secondary attack rate is how many household members are going to get infected when that person is sick. This is a useful parameter because, for instance, if everybody's vaccinated in one family, the chances are much lower, right? Secondary attack rate is lower. So what I'm going to be focusing on is basic reproduction number, which is not a measurement, but a larger, but a model. Okay? So it's modeled, and it demonstrates the meaning of those numbers that you see here in this column, is how many people are going to be infected by a sick person for the duration of their disease. So if you look at Ebola virus, it's really not that contagious. One person who has Ebola virus, on average, would infect about 2 people. Okay? And then you look at measles, which is the most contagious disease known so far to humanity. The measles outbreak in I don't remember where in Minnesota, but it's definitely in Minnesota. There's a big Ethiopian community there, and kids were unvaccinated, and they returned from Ethiopia. So one kid got sick, another kid that got sick after, and it was established that it was from that child, the first sick child. He was lived in the apartments 1 floor above. The virus went through the ventilation. Yeah. Right now, if I have measles and you're unvaccinated, you're all infected. It is the most infectious. However, this is what I want to highlight when you look at this basic reproduction number. First of all, it is not a measure of how bad the disease is. If somebody would tell me Ebola or measles, I pick measles, even if I'm not vaccinated. Okay? Another thing, consider factors that contribute to the spread, i.e. human behavior. So imagine I get infected with measles here in Cleveland. I jump in my car and drive to my ranch in North Dakota. I get out of the car, chances of meeting a person in North Dakota are close to 0 because it's in North Dakota. So I'm not going to infect anyone. Now, there was a real case outbreak during the 2014 Ebola outbreak in Western Africa when a dude died, and during funerals, they didn't give the body to the government as government required. They decided to bury the dude themselves, so they held funerals, and all 100 people that were invited kissed the deceased on the head. Great idea. So, yeah, like if I'm walking around stabbing everybody with a syringe that contains HIV, that's a pretty good transmission rate. Okay? Question?
So it does take into account
Behavior, no. Like if a person behaves, and think about this, the basic reproduction number for SARS-CoV-2, the coronavirus that caused recent pandemic, is estimated to be between 3.5 and 4. Okay? But if I'm not leaving my house and wear a mask, well, first of all, I'm an idiot, but also I'm not going to transmit it to anyone. That make sense? So the goal of epidemic control is to bring a basic reproduction number below 1. Because then the outbreak dies out. Okay? Now, different sources of infection. I'm going to add some comments. Community acquired. What does that mean? What do you think?
hospital acquired,
No. Community acquired.
community acquired,
Here. That's community acquired. Yes. School or nosocomial or hospital acquired. Do you have to be a patient? No. No, you don't. There were cases of visitors. Okay? Now, iatrogenic, you do have to be a patient. Iatrogenic infection is the result of a medical procedure. One of my favorite cases of iatrogenic infection was when a guy got killed in a motorcycle accident. He was an organ donor, and his organs made into a bunch of folks. Well, turns out he was in the incubation period of the rabies virus. And guess what? People who receive the transplant get immunosuppressive treatment. So 7 people died of rabies because they didn't, at the time, they didn't test. Now every transplant donor is tested for rabies. Well, when they deceased, because rabies is uniformly lethal. Like, if you have rabies, you're going to die, period. Like, okay? And finally, zoonotic. That's acquired from animals. Now, can you get infected with SARS-CoV-2, with the coronavirus, in school? It's a community acquired. Can you get infected in a hospital? Yeah, it's a nosocomial. Can you get infected from an animal? Yeah. So my point is, the same pathogen can be transmitted from different sources. That's it. Now, this is just an illustration of our progress in hospital-acquired infections. I'm not going to get into the details. We're doing good. Less and less. Well, pathogenicity and virulence. So pathogenicity, again, if you put 2 epidemiologists in the room and ask them a question, you're going to have not 3, but probably 11 answers. Pathogenicity, we are going to consider this as the ability to cause the disease, period. So let me ask you this. Ebola virus, can it cause a disease? Streptococcus pneumonia, can it cause a disease? Yeah, they're both pathogenic. Now, if somebody would ask me, choose one, it would be strep pneumonia. Because Ebola is much more virulent. Virulence is the comparative parameter that shows how severe the disease is. Does that make sense? So the virulence has 2 sides that determine how bad the disease is. One side is the infectious agent, which may produce different virulence factors. Another side is the host. So let's take a look at the infectious agent side. They can produce virulence factors. Now, remember the very first unit where we're talking about capsule? What are the functions of the capsule, if anyone remembers? From phagocytosis, yes. And the second function? It's sticky, right? Sticky. So if a microorganism, a bacteria, are more sticky, they are better at causing the disease. Would you agree? And if your immune system has troubles in clearing that, it's also not good, right? So that's an example. I'm giving you an example of a virulence factor. Then there's a portal of entry. If I will drink a glass of HIV, am I going to get HIV? No. Unless I have, seriously, unless I have bleeding wounds in the mouth, but let's say I don't. So yeah, I'm fine. Does that make sense? My point is, a microorganism that chooses, quote unquote, a wrong portal of entry may not be virulent. And there can be another aspect. So if somebody would tell me, hey, we have a little drop of liquid that contains 1 million cells of E. coli. We're giving you a choice. You swallow it, or we're going to inject it in your urethra. It will be immediately swallowing because urethra, let's say the injection is painless. Like, it's not about pain. It's about where do you place it. You see what I'm saying? So all those Enterobacteriaceae, if they get into your intestines, it's every man's guess, you know? You have great competition there. You place it in the urinary tract, it's a shit show. Make sense? Or you somehow spray it in the lungs, it's a completely different story. So again, Staph aureus on the skin, okay, you've got impetigo, what a big deal. If it manages to get in the blood, you're going to get endocarditis pretty quickly. So different portals of entry will determine how bad, how horrible the disease will be. And then this. It's in the bottom because scientists, epidemiologists, infectious disease specialists do not agree on that. Some say that the greater the infectious dose, the worse the disease. Others say that's not true. There's no data. And technically they're right. There's no solid data on this. It seems in animal studies that the greater infectious dose will cause more death. But not super conclusive. I know, it's kind of counterintuitive. Yeah. But, and in humans it's really hard to do it. Now, what about host? Well, genetic susceptibility. Some people, one of my favorite examples, plague. There were 3 big pandemics, plague pandemics. One in the 14th century, I don't count the Byzantine in like 600s, 7th century. So one big in 14th century, then one in 17th, give or take, and then early 19th. Guess what? The mortality progressively decreased because people who were more susceptible died. Okay, so genetic selection. Immunosuppression, duh. Pregnancy, complicated. But generally, yes, pregnant, let me put it this way. Infection in pregnant women significantly increases the risks of fetal complications. Not as much for women, but fetal complications, sure. Systemic diseases, obviously, you know, somebody has diabetes, it's not good. Age, It's interesting because it works differently in different populations. I tracked pretty closely the case fatality rate during COVID pandemic. It went exponentially higher with age. So, like, state of Ohio had 2 pediatric deaths. I think it was under 12, something like that. 2 for the entire length of pandemic. The number of people over 80, like if you get in the hospital, you're not walking out. Okay, you have very low chances. So age, other infections. If you have one, another, doesn't make it better. Trauma surgery, immunosuppression, specific community. So, are you familiar with the Spanish flu in 1918? Yeah? Well, first of all, it didn't start in Spain. Most likely started in the US. But do you know the age group that got absolutely hammered? Interestingly enough, no, 20 to 40. Because older people and kids both had a very specific exposures. There were other flu outbreaks before that made them partially immune to that specific strain. Okay, and that was just they were fine. Okay. So why scientists are so skeptical about the dose thing? So first of all, let's define median infectious and median lethal dose. Do you need any explanation of the difference between infectious and lethal? I don't think so. Right? But what about median? So median means that the half of the infected group median lethal means half of the infected group dies. Median infectious dose means that half of the group actually becomes infected. Okay? So this table compares median infectious dose for different pathogens. Okay? And I want to compare. And hear me out. First of all, broadly speaking, would you agree that the pathogen that has a smaller median infectious dose seems more dangerous? Give it a thought. So think of it this way. You need one particle of a norovirus to develop the disease. You need 200 bacterial cells of E. coli to develop the disease, right? On the first glance, norovirus seems much worse because you need just one tiny particle, right? Remember, infectious doses cannot be compared between different microorganisms. They do not reflect virulence of different microorganisms. I'm talking about different species or phyla or whatever. However, within the same species, when we talk about different strains, look at E. coli. So the bottom one is enterotoxigenic, which causes diarrhea, period. Then you have enteropathogenic, also causes diarrhea. And then you have an enteroinvasive. That's a pretty severe infection of the colon that can result in colon perforation. And on top of it, you can see enterohemorrhagic E. coli, the H7O157. Explain why this is relevant to your life. Lowest infectious dose, right? This is the scariest. So within the same species, within the very closely related group, you can say that lower infectious dose is associated with greater virulence. You cannot make this comparison between different groups. Okay? All good? And I just want to emphasize, folks, determining the infectious dose in humans is super hard. Because for this we need people, we need to infect people. And it's not ethical. Okay, portals of entry and exit. Just understand how we can get microbes. Like, respiratory, fecal-oral, contact, blood transfusion, that kind of stuff. Do not memorize the examples. Like, this shouldn't be a separate slide. The only thing on this slide that I want you to seriously memorize is that acronym. First of all, you're definitely going to see it if you go into nursing or any other like medical profession associated with broad spectrum care. So TORCH stands for toxoplasma. Then you have wonderful other, rubella CMV, cytomegalovirus, and herpes simplex. Those are pathogens that can be transmitted via placenta. That make sense? Now, another point that I wanted to make. Scientists are really bad at acronyms. Like having O that can cover all types of scenes. And to my knowledge, American Society of Microbiology has sort of a competition open. If you can offer a good acronym that would take into account all the pathogens. Because it's not only this, it's syphilis, hepatitis B, chickenpox, HIV, parvovirus, erythema infectiosum. So now let's talk about stages of infection. So first, microorganisms must adhere. I can ask you about the 5 mechanisms of bacterial adhesion. We talked about those structures when we covered bacteria. The only difference is adhesins. Those are proteins on the surface. Okay? Then we have viruses. Every virus, and I think I made this point when we were talking about herpes, must interact with a receptor. If there's no receptor, no interaction, no infection. One of my favorite examples, polio. It infects humans and apes, not good at infecting other monkeys, and does not infect other animals because there's no receptor in them. And hepatitis C, which infects humans and apes, and to my knowledge, you can't work with apes anymore. Banned because of the ethical. Like you can work on monkeys, like basically we're saying that there are better and worse monkeys. Like, yeah, you can like lousy monkeys are okay to kill. Okay? So this is why receptors can define which organisms can be infected. So for instance, that hepatitis C, pretty much human infection. West Nile virus is one of my favorites. There is a term, it's called promiscuous virus. It infects, to my knowledge, every vertebrate and a bunch of invertebrates. It doesn't do it in nature. Normally, you can stick a needle with this virus into a snake and it's going to get infected. Okay? Now, invasion. So I'm not going to ask you anything in details about invasion. I just want you to understand that the infection can be outside of the cell by bacteria, for instance, and sometimes can be inside of the cell. It can be bacteria. All viruses are intracellular pathogens. Okay? And this brings us to the virulence factors. I think what we're going to do the stages of infection, wrap it up and go to the lab. Okay? So basically just virulence factors. So virulence factors that I will discuss here include exoenzymes and toxins. So let's start with exoenzymes. What does it mean, exoenzymes? When microorganism produces them, these enzymes, where do they go? Inside of the cell, right? So they can destroy tissues. For instance, hyaluronidase, produced by various species of Staphylococcus, can destroy extracellular matrix and result in tissues falling apart. Or coagulase, produced by Staphylococci, those are sneaky little bastards. Coagulase stimulates formation of fibrin, which forms protective mesh around bacteria so macrophages cannot reach. Okay? That makes sense. The actions of exoenzymes. Then we have cytokine storm. Did we talk about influenza and snot chase? Well, for viruses, it's not going to be exoenzyme. It's just one of my favorite stories. So influenza virus, when it infects human cells, it needs to shut down cellular protein synthesis and establish the synthesis of its own genes. Now, if you remember transcription and translation, mostly translation, in order for our cells to translate a message to make a protein, they have to recognize 5' cap structure on RNA. So what virus does, influenza virus does, it takes that cap and puts it back on its own RNA, and it's called cap snatching. And the enzyme that does it is snot chase. It's a name, which I find extremely funny on many levels. Now, viruses often cause what's called a cytokine storm. So, when you get, let's say when you get influenza, your immune system starts fighting infected cells, killing infected cells. And during that fight, it releases a bunch of inflammatory cytokines. These cytokines trigger all those responses that you feel. Myalgia, arthralgia, headache, fever, all of that is your own immune system releasing too much cytokines. Okay? So, for instance, in hemorrhagic fevers such as Ebola or Marburg or Lassa or Nipah, you name it, people have all the symptoms, hemorrhages and stuff, not because of the virus itself, but because of your immune system going completely berserk on your own tissues. And it's been shown that actually immunosuppressive therapies help. For what it's worth, when people have COVID, if they end up in the hospital with respiratory distress and hypoxygenation, the first treatment that is given to them is what? Anyone knows? Well, obviously, nasal cannula with oxygen, yes, so they don't die, but that hyperinflammatory state is treated with one of the cheapest, yes, steroids. And it is really effective. It basically is down the cytokine storm. So what do you need to know about exoenzymes? These 4 categories and what they do. So yeah, we're going to do exoenzymes in pause. So glycohydrolases break down carbohydrates. Nucleases break down DNA. Phospholipases, well, fair guess, phospholipids. And collagenases, well, proteases, I'm sorry, break down proteins. Honestly, a question of taking candy from a baby. Like, the answer is pretty much in the name. So know these 4 classes of exoenzymes. Sounds good? So, since we have some stuff to do, let's wrap it up for today. So, just a reminder, we started talking about different virulence factors, and so far we went over exoenzymes. Now we have to talk about the toxins. So in this conversation, I want you to focus on sort of the differences between the endotoxins and exotoxins. So let's start with which microorganisms can generate either of those. So endotoxins, which we encountered before, the term lipopolysaccharide, are found only in the outer membrane of the gram-negative microorganisms. Now, exotoxins, molecules that are released by the bacterial cells into the environment, can be found in actually all of them, gram-positive or gram-negative. So the composition of endotoxin is pretty standard among gram-negatives. It's a lipid component of lipopolysaccharide that acts as a toxin. So what it means for us is that if you take several different gram-negative microorganisms, let's say Pseudomonas, Enterobacter, Salmonella, Bacteroides, Parabacteroides, they're all going to have pretty much the same endotoxin in the outer membrane. The structure of the exotoxins, these guys are overwhelmingly proteins of different shape and function. So the effects that endotoxins and exotoxins have on the host also vary dramatically. So lipopolysaccharide, the LPS, right? It causes a general stimulation of your innate immunity for the most part, which results in inflammation and fever. You may experience headaches, muscle aches, joint aches. You get the gist, right? And again, the point is that your immune response to endotoxin from Salmonella and endotoxin from Pseudomonas will be pretty much the same. So the symptoms are going to be very similar. On the contrary, exotoxins incur very specific damage to the cells. Okay, they have different mechanisms of action, which we will generally cover. And the effect of each individual exotoxin can vary from its counterparts. Endotoxin is heat stable, and that poses a very serious problem. Think about this. Let's say you have a solution, like a watery solution, right, that you want to inject into person. So you autoclave this solution. Let's say it's, heat stable. Okay, you autoclave this solution to kill all the microorganisms. You kill all the microorganisms. The only problem is that endotoxin will be released from dead bacterial cells, and when you inject this solution into a patient will produce the response to endotoxin, although there are no bacteria left. Now, exotoxins, most are heat labile, which means that they, will be inactivated by the heat. Now, we had a conversation about median lethal dose. For endotoxin, it's really high. Like, you have to really, load up a patient with this. For exotoxins, it's very low, and we can discuss how low that is. And the last bit, which is kind of important, endotoxin does not stimulate specific immunity, adaptive immunity, so there are no antibodies. So every exposure to endotoxin will be taken by your immune system as a new one. On the contrary, exotoxins, I may have misspoke calling the, endotoxins do not stimulate immunity. Exotoxins do. You know the term tetanus shot, right? So I'm not referring to the vaccine against tetanus. I'm referring to the shot that you receive after you have been potentially exposed to the microorganism. This tetanus shot is antibodies against the tetanosspasmin, that are aimed to block its action and prevent you from developing the tetanus in the first place. Now, some terms to know. Toxicity, ability to produce toxins. Toxinosis, a disease. Toxemia, a toxin in the blood. And intoxication, the ingestion of the toxin. One may ask whether or not they should know anything about LAL test. That big paragraph that I haven't highlighted yet. I mean, should you? Yes. Will I ask this on the exam? No. The whole story about LAL test was a bit of my personal capricious decision. So we're going to skip LAL test and jump straight to this terrifying table from which you only need to know 3 categories of toxins. First category is intracellular targeting toxins. What do you need to know? These toxins target specific cellular processes. Okay? Look at the first example. The cholera toxin activates the enzyme adenylate cyclase in the epithelial cells in the intestine. Okay? Like, it's very specific. Am I making sense? Or look at diphtheria toxin, which inhibits protein synthesis by inhibiting certain translation factor. Sounds good? Second category that you need to know is membrane disrupting toxins. The name speaks for itself. These toxins disrupt the membrane. And finally, superantigens. We mentioned those guys before. So superantigens overstimulate the immune system, and that results in the cytokine storm and potential death, basically septic response. You on the same page so far, everybody? So again, can I ask you what is the mechanism of action of tetanus toxin? No. Can I describe the mechanism of action of tetanus toxin and ask which category it belongs to? Yes. Clear? So don't memorize examples, much rather read through them so you have a picture, you have like understanding of what these toxins do. Not surprisingly, that microorganisms have to evade the immunity. So, as always, do you need to memorize examples? No. But you must match the mechanism and its general description. So, we're going to start with what you already know, the capsule. The capsule, as you know, prevents phagocytosis of encapsulated cell, right? Macrophages cannot eat it. Some bacteria release proteases. So these enzymes specifically targeted at the antibodies. So here you can see the broken antibody. Intracellular infection is the kind of immune evasion strategy as well. If you can survive in the macrophage, how can macrophage eat you? It already did and you're still alive. A lot of microorganisms, I want to say mostly viruses, but it wouldn't be true. Almost all microorganisms have some kind of the strategy to inhibit your innate immune responses, such as an interferon pathway. In my opinion, one of the coolest mechanisms of immune evasion, and I want to explain it because you're probably going to be amazed too. It's called molecular mimicry. So there's this this helminth, Schistosoma mancini. It's a very small worm that ends up in your bloodstream, in the circulation. We'll talk to it in Unit 6, but just believe me, it ends up, it sits in the capillaries that surround either your intestines or your urinary bladder, okay? Small, called blood fluke. And it covers itself, physically covers its surface with human proteins. So the immune system sees it and says, yeah, it's fine. Those are human proteins, okay? That's pretty cool. Yeah, pretty ingenious, huh?
They're genius. But
Yeah, yeah. So that's molecular mimicry. I put it in a separate category, complement inhibition. A lot of organisms, particularly bacteria, love inhibit complement, one of the components of the innate immune response. And finally, very specific mechanisms, killing white blood cells, killing leukocytes. Staphylococci and streptococci produce proteins called leukocidins that can destroy those white blood cells, okay? Now, when I go over these notes, I tell students that this single slide, one slide, can be developed into 3 credit hour, 1 semester course. There are so many mechanisms and they're so interesting. But one idea I want to kind of float in front of you is so-called Red Queen paradox. I didn't read the Alice Through the Looking Glass, the book by Lewis Carroll, but I have a pretty good idea what's going on. And there's a conversation between one of the characters, the Red Queen, and Alice. They try to make it to a train station. And they run to a train station and they don't seem to get any closer to it. And Alice is being frustrated and says that in our world, when we run somewhere, we make it there. And Red Queen responds that in our world, you have to run as fast as you can just to stay in one place. This reflects the idea of coevolution of pathogens and their hosts. I'm not asking, I'm just stating, some of you may have had cold sores. Some of you may have had chicken pox. It kind of sucked, but not as much as, let's say, a case of coronavirus or a new pandemic strain of flu. I'm not even mentioning such infections as Nipah or Marburg or Lissa viruses, different hemorrhagic fevers. So when you start thinking about that, herpes viruses such as chickenpox and herpes simplex, they coexisted with humans, and it's been shown before we were humans. They were infecting our ancestors. Over the years, they learned not to kill us, and they also learned how to trick our immune system so it doesn't kill them. Make sense? So basically, the selective pressure of our immune system selected for the sneaky ones, but the sneaky ones have no quote-unquote intention of killing the host, because why? And it generally is, with exceptions, generally sort of a rule that this is the constant running in one place between the host and the infectious agent. So the immune system tries to eliminate it, the infectious agent tries to not get eliminated, and the most successful strategy wins, and usually it is attenuation of the pathogen. Does that make sense? So here we get into the stages of disease. Stages are right, the graph is wrong. I will explain why. So first stage is in incubation. You don't have signs or symptoms. That's a definition. You shouldn't have. Then you may have prodromal stage. So as an example, I'm going to use influenza. You know when you start feeling crummy, but you're still kind of okay-ish? That's prodromal. It's really hard to define. So you may have like a scratchy throat or something like that. And then you enter the full illness phase. So on this graph, the phases are highlighted in blue, the blue-colored line. So illness is your severe symptoms. This is when you sit at home or lay down and you have everything hurts, you feel like crap, you have high fever and so on and so forth. So this is illness, right? So you get to that peak right here. What can happen to you even if you have a flu? What are the two possible outcomes? One option is what? Can you recover from flu? No, you can't. In some cases you can't. So two outcomes. Either you survive or you die. So if you die, we stop here. If you don't die, symptoms start going away. Okay? So this is a decline phase of the disease. And then you have convalescent phase. So convalescent phase is usually defined as you kind of feel okay, but there are some lingering consequences. Different diseases behave differently. We're going to talk about it in a bit, okay? But like, I'm going to give you an example which I hope will turn you off of restaurants, so there will be more space for me. Are you familiar with norovirus? The two-bucket disease, yeah? So it turns out norovirus can be excreted in feces up to 2 weeks after the resolution of symptoms. So this is what I want you to think about next time you are in the restaurant and the waiter brings you food. Just ask yourself, maybe this guy or gal just got better from norovirus, but those viruses still coming out. Maybe they are on their hands. Okay? That is true, actually. So convalescent, practically gone. Symptoms, I mean. Now, why did I say that this graph is wrong? So this graph shows you that the clinical manifestation corresponds to the number of infectious particles, right? It's not true. Just, you can't make this generalization. Sometimes it does, sometimes it doesn't. If we take disease such as Ebola or very first coronavirus that caused an outbreak, pretty much pandemic in 2003, that would be true. People who transmit those viruses most actively are the people who are most sick. But if you look at the transmission of influenza or SARS-CoV-2, for influenza it is known that the peak viral replication is on the first day of symptoms. Then it starts to decline actually. For SARS-CoV-2, the peak is 1 day before the symptoms and the day of symptoms. So if somebody has been sick for, let's say, 1 week, they shed almost no virus. They're not particularly infectious. Does that make sense, y'all? Okay. Now, we characterize the disease based on the signs and symptoms. So what is the difference between the two? Well, it turns out signs are objective, symptoms are subjective. So you are a bunch of physicians, I'm your patient, tell you that my tummy hurts. Sign or symptom? It's a symptom because it's subjective. I tell you, I always give an example. My son had some intestinal infection and I asked him, your stomach pain on the scale from 0 to 10, where are you at? And he goes, 9. And I go, 9? At 9, like I'm writing my last my will. Like, that's 9. I'm dead. It's perception. Okay? Now, I'm in front of you and I barf. That vomiting, sign or symptom? If I tell you that I'm nauseous, symptom. So you have to be careful. Sign does not have to be quantitative, but it has to be objective. Okay? One of the very interesting recent developments that I learned about. Turns out, you're familiar with the huge outbreak of measles that is going on now in both countries? Great job, I'm not blaming you, but great job. We brought back the disease that was eliminated in the United States in 2003. Turns out about a third of measles cases present without rash. This is bad, because you have a garden variety respiratory infection then with just cough and fever and, that's it. But quarantine requirements for measles are much stricter than for any other respiratory disease. Okay. Localization. This is interesting and boring at the same time because we're just going to be talking about a bunch of terms. Localized infection. Portal of entry. You have strep throat. That's localized. Okay. Focal infection. Spreads from the original site to someplace else. Your Streptococcus pyogenes went from the throat to the heart, caused endocarditis. That's your focal endocarditis. Systemic infection, agents everywhere. Carried around by the blood. Okay? The term that ends with emia means the microbe is in the blood. So, viremia, bacteremia, parasitemia, and so on. The term septicemia usually is applied to a specific process of the replication of bacteria in the blood. Does that make sense, y'all? Yeah? Just replication. Not just presence, but replication. Primary versus secondary. It is known that influenza virus infection increases the chances of streptococcal pneumonia. So, primary happens first, and then increases the chances of getting the second. Okay? Now, one term that I don't have here, but I'm going to put it on the screen. Acute infection. Short. Doesn't refer to the severity, just short. When you get sick with noro, it usually lasts for 48 hours. That's an acute infection. Cool? The opposite of acute is chronic. You get a long-term infection which has a clinical syndrome, like hepatitis C, or giardia lamblia, which gives people 4 to 5 months of steatorrhea, which kind of sucks. Then there's a latent infection. We talked about it in the context of herpes. Remember, dormant state. So I want you to be very clear about that. During the latent infection, in the period of latency, infectious agent does not replicate. There are no symptoms. It can reactivate from time to time, but other than that, doesn't replicate. Kind of opposite is a persistent infection. Did I mention the joke about 2 epidemiologists in one room? You put 2 epidemiologists in one room, ask them the question, you get 5 answers. So, the only reason why I use persistent infection to specifically describe a very unique situation, when you have a reproduction of microorganisms without clinical symptom, potentially pathogenic microorganisms. Now, I'm going to use BK virus and CMV. So cytomegalovirus, which we mentioned before, especially in kids, when they get infected, they often develop a very interesting type of infection when they have no clinical symptoms whatsoever, no detectable pathologies, but they shed huge amounts of virus in the urine. So it replicates. Another example is BK virus, which infects a very large number of people, kidneys, and causes no symptoms if a person is immunocompetent. When the person becomes severely immunocompromised, that's a different story. It basically kills the kidneys. But in pregnant women who are mildly immunocompromised, BK virus reproduces, gets shed in the urine, but causes no observable pathology whatsoever. So that's your persistent replication without doing anything to you. Okay? Now, the term sequelae, long-term, often permanent damage to tissues and organs. Remember when COVID just started, there were some voices suggesting to, let it rip through the population. I'm not talking about the fact that at the time, case fatality ratio in the States was 2%. So for the country that has 330 million people, we're talking 6.5 million deaths. That's a separate story. Do you know anyone who has long COVID? Have you heard the term? So there are millions of people in the US who have serious, real complications of the disease that they got back then, when there was no vaccine, and you have to go to work and stuff like that. And they still suffer, and there's no treatment really. We don't really know what to do. So that's to the idea of when we're dealing with something that we don't know, it's a lot of hubris to presume that, oh yeah, it's going to be fine. Yeah, maybe it won't. Okay? Maybe it won't. And it wasn't. So this is why I wanted to plug it in. That's a long-term sequelae. Okay? It's a great, when a person like me tells you it's a great question, you know what it means? I have no fucking idea. I did not see any data on whether the risk of long COVID changes with age. Generally, remember COVID was worse in older people. I would say that, kids had lower chances to get severe disease, and that is why there's less long COVID in kids. But if you look at very old people, there's not a lot of long COVID. You know why? Because they died. Okay? So you have to take into account, is it age-related, or is it just because they either didn't have a very severe infection or had super severe infection? Okay? That makes sense? So, portals of exit. Boring, it should have been called vectors and reservoirs. Microorganism can leave human body via multiple routes. That's what I want to say. It can be fecal, it can be mucosal secretions, it can be urinary, respiratory, contact, whatever, blood. And this brings us to the conversation about the reservoir and the host. Okay? The difference between the two is very shaky. Generally, it is accepted that a reservoir is something without which microorganism cannot exist. Understood? Here's an example. Measles. Measles is a virus, so it requires a host. It requires a living organism, okay? The only living organism in which measles reproduces are humans. Humans are a reservoir. How do we know? If tomorrow humans disappear, the virus disappears. Clear? I'm trying to think of another. Okay. The reservoir for brain-eating amoeba Naegleria fowleri is water and soil. If tomorrow we somehow make water and soil in the entire world disappear, we can guarantee this amoeba disappears. Is that clear? Host is not required. So here's an example. Rabies. It has multiple reservoirs, such as bats, skunks, opossums, foxes, okay? Some of them get sick and die, some of them don't. Bats do not get sick from rabies, they just carry it around. Now, usually when we consider infection in the disease, we say, oh, it is in the host. So like when a human gets sick with rabies, right? We are the host, but we're not reservoir. Am I making sense? For measles, we are both. For coronavirus, SARS-CoV-2, we are one of the many reservoirs because it persists in humans, in deer, in cats, in dogs, in mink, you name it. Does it make sense, y'all? Okay. Now, vector. Arthropod, go ahead. That's a good question. So I tend to use these two terms to separate between just carrying and being sick. So and again, there are multiple interpretations. Are we the reservoir for measles? Yes. Are we the hosts? Yes. Bats that carry around rabies, they don't appear to be sick at all. Can we consider them a host? I wouldn't, because the term that serves better is a reservoir. Okay, here's an example. The virus with a reservoir that is completely different from the host, polio. The reservoir is soil. We are the hosts. We're getting sick. Okay? Like tomorrow you remove all the soil, it's gone. So let's go back to vectors. There are two types, biological and mechanical. I'm going to start with mechanical. Here's the fly. Where's the fly? Here's the fly. The fly that sat on the pile of poop and then sat on the sandwich and mechanically transmitted microorganisms from poop on the sandwich. Now, this is the biological vector. So you see this mosquito takes a blood meal and it ingests the blood with the pathogen in it, such as yellow fever or malaria. That would be mosquitoes, ticks, midges, things like that, okay? That would be a vector. I have been studying vector-borne diseases for quite a lot of time, and I have a particular aversion to them. I love them. Okay. Now, transmission routes and carriers. So let's start with the carriers. The carrier, human sources of infection. Okay. So asymptomatic, no signs or symptoms, and will never develop them. Am I clear? That's the difference from incubation, which has the infection, will get sick, but right now it's in incubation. So one of the biggest problems with SARS-CoV-2, the spread, was that it was very easily transmitted by people who are asymptomatic or in the incubation period. So it was established that 80% of the transmission is carried out by 20% of infected people. So there were super spreaders, probably because they were asymptomatic. Like maybe I have replicating SARS-CoV-2 in me right now, but I feel fine. And if you would tell me, hey, let's go to get a pint at a pub, I would go and get a pint. Okay? And I will infect a bunch of people. And it's not my fault because I don't know. Make sense? So that's asymptomatic and incubation. Then we have convalescent, that norovirus. And I have another good news for you. It can be transmitted not only in the convalescent phase, it can be easily transmitted during incubation phase. So a person can be infectious, contagious, for like easily 3 weeks. Okay, imagine how many people it can infect. Okay, question, Steven?
Okay, so you're not symptomatic in the convalescence? Like you're better, but you still shed virus?
Yeah.
Okay.
Yes. Yes. If you plan to travel in, I'm going to call it South and Southeast Asia. So basically starting at India and all the way to the east, there's a good chance, because of the local microbial populations, to catch salmonella. If you catch salmonella and you completely recover, you're going to shed it for 4 more weeks after you return home. So, chronic, obviously, chronically infected, passive. A physician who wears a tie. A tie touches one patient, then touches another patient, and they lean over. That's your transmission. Does that make sense? Like a nurse who doesn't follow the protocol, something like that. Inanimate sources, vehicles and fomites. Vehicles, food and water. That's it. Fomites, inanimate objects. So if I have flu and I sneeze in my hand and touch the doorknob, and then you touch the doorknob and get infected, doorknob served as a fomite. There were cases of cattle-adapted bird flu transmitted to humans with milk. So a cow is infected, they milk the cow, drink milk, get the bird flu. The milk would be a vehicle in this case. And this brings us to direct versus indirect routes of transmission. So direct routes, let's explain what it means. Respiratory, that refers to the situation when the transmission is promulgated by your respiratory activities. Is breathing a respiratory activity? So that's one. Speaking, singing, coughing, sneezing, anything basically that comes out of your respiratory system and forms droplets of different sizes is considered to be respiratory transmission. Okay? 8 out of, so when I want you to understand that when we talk about respiratory transmission, let's use influenza. Okay, it's fine. When you cough or sneeze or breathe, you're not expelling virus particles. You're expelling small water droplets, fluid droplets. Does that make sense? That carry those particles inside them. So this is why masks work, because they absorb at least large particles, okay, and reduce the chances of being infected. Now that question, 8 out of 10. When is respiratory transmission more efficient? I'm going to make it 9 out of 10. During cold winter or that type, okay, I'm going to dial it down to 8 out of 10. What is better for respiratory transmission? Warm and humid or cold and dry?
Warm and humid.
Exactly. Because particles, even not necessarily cold and dry, just dry. Because when it's dry, those droplets evaporate. When it's not, they don't. So contact. Anything from sexual to skin to vomitus, you name it. Okay? Vertical transmission, mother to child. Can be congenital, can be transplacental or birth canal or breast milk. That's all considered vertical. Vector transmission. Now, not every vector transmitted disease can be transmitted directly. Have you heard about why West? No, have you heard about Lyme disease? Yes? Yes? No? No, I envy you if you haven't. But it's a disease. What transmits Lyme disease? Ticks. Ticks collected on deer or rodents and then transmitted to humans. Can a tick bite you, suck your blood, fall off, bite another person, and transmit your Lyme disease to them? No. It doesn't go high enough in numbers in the blood. So there's not enough bacteria in your blood to transmit it to somebody else. So this is an example. Yeah, it's a vector borne, but it can't be transmitted from person to person. Diseases like dengue virus, yellow fever virus, malaria can be transmitted from person to person by mosquitoes, by a vector. Is that clear? Indirect transmission. So fomites, that's we already discussed. Vehicle, and I put air as a separate modality of transmission. So this happens when you form. I can give you a couple of examples. One example is Microbiome avium. We talked about it. So it's in the feces of birds. Okay? You have to kind of actively aerosolize bird poop to get infected. And you can't really transmit it to another person. You have to get it from the bird poop. Am I making sense? We talked about histoplasma capsulatum, same story. Not from person to person, but you get it from bird and bat poop. Another example that I have here is hantavirus. Are you familiar with the Four Corners region in the United States? There are 4 states forming like 4 corners come together. I think it's New Mexico, Arizona, Nevada, and Utah. Maybe, I don't remember the 4th. Anyway, so there was an outbreak of a virus that was transmitted to people because deer, mice, were getting in the houses in search for food and pooped on the floor, and people swept the floors and aerosolized the virus, got infected, severe respiratory distress. Okay? Not transmitted from person to person. That's an example of air transmission. A neat detail about that story is that initially the virus was named Four Corners virus, which, reasonable, right? Now imagine you're a tourist and you want to visit this Four Corners region, and then you hear that there's a deadly Four Corners virus that kills almost everyone. Would you go? Probably not. So people who lived there sent a mail to the nerds that name viruses. There's a group of nerds that does it, asking to change the name. They couldn't come up with anything else. Anyone speak Spanish? They named the virus Sin Nombre. What does that mean? No name. They named it No Name Virus, Sin Nombre virus. Okay? Which I think is kind of we're not going to give it any name. So, let's talk about some fun stuff. So we're going to talk about a couple of vector-borne parasitic diseases caused by the members of the genus Trypanosoma. First one is the African sleeping sickness. It is transmitted by tsetse flies, which are, I never saw one in real life, and I'm very happy about that. They're pretty big, And when they take a blood meal, they inject this microorganism here, Trypanosoma brucei, inject these microorganisms in the blood, and it spreads through the blood and infects almost all organs. And it's a pretty long disease. Familiar, Richard? Yeah. So it's a long infection. If it's left untreated, it's uniformly lethal. Can be treated. Okay, so basically it's called sleeping sickness because there's sleep disturbances. What's interesting about it, if you ever donated blood in the United States, one of the questions on the questionnaire is, have you ever was diagnosed with African sleeping sickness? If the answer is yes, you deferred for life because you consider to be positive for the rest of your life, because you can't confirm that all microorganisms were eliminated. Okay? Now, epidemiologically, sub-Saharan Africa. So this map here, sub-Saharan Africa. Okay? Now, Richard may correct me, should correct me, but I believe it is transmitted from the cattle, like buffaloes, and one of the main determinant of the presence of those flies is the proximity of water. So you can find like regions, and I'm talking about the stuff that I've never seen in my life, regions where people like don't build houses close to water because they know they're going to get it. Okay. Now, what's interesting is that there's another disease caused by a different Trypanosoma cruzi, which is present. Trypanosoma brucei, causative agent of African sleeping sickness, is only in Africa. Period. That's it. Trypanosoma cruzi is only in the South and Central America. To be honest, transmission route is much cooler. So the kissing bug, from I think the family Tratominae, another term for it is reduvid bug, lives in the thatched roofs of rural houses, not like that, not like, the proper ceiling, but hay and leaves and, sticks and stuff. During the night, it's a feeding time, it lands on human face and bites either the corner of the mouth or the lateral commissure of the eye, because the skin is kind of thin. The bite is very, very small. It's a surgical incision, takes a blood meal, shits in the wound, and leaves. When person wakes up, the poop, the bug poop, irritates the wound, so the person starts scratching it, effectively rubbing trypanosoma into the blood. Okay, so like this kid here, you see the enlarged cheek? That's a Romano's sign. Means that this kid was bitten into that right lateral commissure. So then microorganism spreads pretty much just like another trypanosoma, spreads systemically. Your spleen tries to get rid of it, therefore splenomegaly. It also very happily infects liver, so hepatomegaly. That's not the end of the story though, because Trypanosoma cruzi loves muscles, specifically smooth and cardiac muscles, and it infects them. If it infects a cardiac muscle, it causes inflammation, which results in the damage to cardiac muscle and myopathy. So what's important for you to understand is that it's for life. Even if you kind of treat, the heart's gone. Okay? Other two kind of cool symptoms, megacolon and megaesophagus. So in these cases, the infection of a smooth muscle in the colon or esophagus results in the weakness and lack of peristalsis. If it's esophagus, you can't swallow. If it's colon, You can't poop. Constipation. Okay? Now, just like with African sleeping sickness, treatment is not sterilizing, meaning that you're positive for life. But chances of you encountering a patient with African sleeping sickness, very low. Again, unless you work with a very specific immigrant population. Chances of you encountering someone with Chagas disease, not high, but reasonable, because it's a long disease, and many patients start experiencing symptoms when they're like well in their 40s. So imagine people emigrate in the United States from Central or South America, and here they start experiencing symptoms. Okay? Now, I don't know if you've seen the headlines, but you know, if it bleeds, it leads. So people, like, there are headlines, deadly Chagas disease is making its way into southern United States, which is true. There were cases. But the thing is, we don't live in the huts with thatched roofs. So you really have to get out in the wild to get infected. So few cases, it's not going to be as bad of a problem as it is in the, look, this map, right? You can see that the entire Central and South America is low to high, and Argentina and, what is it, Bolivia, I think? Bolivia? And Paraguay. They affected pretty severely, okay? But there's a special population of patients in America, Texas, Arizona, New Mexico, that have Chagas disease pretty regularly. They have dogs. Yeah? If your dog sees a bug, what does it try to do? Eat it. Eat it. Turns out that's another route of infection. If you eat that bug, you get infected. Now, one may say, well, humans don't eat bugs. Well, not deliberately. There are cases of transmission in Brazil where they make refreshing drink out of sugarcane. They basically run sugarcane through the juicer. Okay? Sometimes there is a triatomine bug among the sugarcane pieces, so they juice it as well. There were cases of transmission through that drink. It's not common, but like it's recommended. Maybe not. So we have a few things to wrap up about epidemiology, and then we're going to give ourselves a well-deserved break. So, Koch's postulates. Let's start with Koch's postulates. So, postulate number 1. The infectious agent must be found only in the sick, but not in a healthy organism. Okay? Kind of makes sense, except for one thing. We have asymptomatic infections. Right? Right? We have people who are asymptomatically infected. And full disclaimer, I'm not shitting on Robert Koch. He's a great microbiologist. I'm just showing you how hard it is to implement those postulates in real life. Then you must isolate a causative agent in a pure culture. Not easy. Well, not always easy. Okay, for instance, Middle Eastern respiratory syndrome, the coronavirus, MERS coronavirus, took quite a while to isolate in pure culture. And Warren and Marshall, it took them 8 months to finally grow Helicobacter pylori. And then the biggest trick, the third postulate. When the agent is injected into susceptible organism, it must cause the same disease. when you have influenza, how you doing? At the end of the day, usually, do you die? Usually not. If you inject mice with influenza, they will probably die. Herpes, we usually survive. You can stick HSV1 in a mouse and it will die. Opposite example. Dengue virus. In humans, causes hemorrhagic fever. You inject it into a mouse, just like in a human, just pretty much mimicking the mosquito. Just take a regular mouse, stick it, let's say, subcutaneously. Instead of hemorrhagic fever, a mouse will develop encephalitis. So my point is, if you isolate it from humans, then sure, go ahead, inject humans. See how long it's going to last before you earn the title, second Mengele, you know? But when you start looking at model organisms, it becomes complicated. Clear? The fourth postulate is kind of obvious. Sort of, the organism dies, you isolate a pathogen, it should be the same. Okay, fine. Koch's postulates are really hard to apply. Okay? Now it's, I forgot the criteria. Shepherd something. There are criteria that allow you to establish the causative relationship. They're more complicated than Koch's postulates, but they were good for its time because he identified cholera, Mycobacterium tuberculosis, and something else, which I don't remember. Like, he linked a disease and a pathogen. Now, what about molecular Koch's postulates? I'm not going to spend a lot of time on this. It shows you the example how it works. Molecular Koch's postulates refer to the method when you identify a specific genetic determinant of a virulence. For instance, enterohemorrhagic coli that causes really severe infection in humans. It's been shown that it produces sugar toxin. When this gene is knocked out, EHEC becomes much less virulent. When you knock it back in, the virulence is restored. So link between the genetics and the virulence is the aim of molecular Koch's postulates. Okay, incidence and prevalence. Finally, I can clear my name. So incidence. I'm going to give you units, okay? Number of cases, usually per year. You can use any time frame. Per year, per 100,000. Now, question. Can you do incidence using a month and 10 people? Sure. It's not going to be as representative, but sure. It's sort of a gold standard, but you can use different measurements of this. Like for very rare diseases, we sometimes report it as number of cases in the US. Clear? Prevalence. Number of cases, we're talking current cases, per 100,000, not per year. So if you will take the entire US population right now and somehow determine who is infected with influenza and who is not, that's going to be the current prevalence. I'm going to ask you a question which is about 5 out of 10. I'm going to give you a hint. Let's say we're talking about respiratory viruses, influenza and SARS-CoV-2, okay? So they infect our throat, cause us to cough, right? But we also swallow the mucus, right? We do it. How can you estimate the prevalence of influenza and SARS-CoV-2 cheaply in the entire United States, even based on counties. Give me an idea what you're going to be testing. Not a hint. What you're going to be testing. We swallow it. Huh? Well, feces is really hard to test individually. Can you kind of get around that? You're on the right track.
Urine?
Huh?
Urine?
No. Again, you can't go around the houses, right? Is there a way to test the county? Go on, I think you have an idea. Sewage. Sewage testing is now ongoing in almost every county in the United States. They test for a variety of infections, and there's when you start seeing the numbers coming up, there's going to be an outbreak. A very cool way. Morbidity. I'm tired of that term. We're not talking about it. It's used so frivolously. Okay. Mortality. Okay, so there are several ways to measure it. Way number one. Number of deaths per year in 100,000 people. Make sense? Option number two is called CFR, which is case fatality ratio. Number of deaths over number of cases. So let's say you have 1,000 people who are infected. 10 of them die. Case fatality ratio is 1%. Sounds good? So I want to bring to your attention the major difference between these two parameters, mortality and CFR. I didn't look up the statistics, but actually have it here. Okay. So let's say the number of cases, number of people who die of norovirus in the United States in a year is approximately 700. Ballpark, give or take 100. Number of people that die of rabies in the United States is in single digits in a year. Okay? So this is an approximation of the mortality parameter, that. So based on this, what is the more serious public health concern? Rabies or norovirus? Noro. It kills, 100 times more. Number of people who get infected with norovirus in the United States yearly is about 170 million. Only 700 of them die. Number of people who are infected with rabies in the United States is the same as the number who die, because there's 100% mortality. So you need to use these parameters appropriately. You see what I'm trying to say? If you want to show public health impact, that's per 100,000. If you want to show how deadly the disease is, that is CFR. Case fatality ratio. Okay? And it's not a perfect measurement, but I don't want to go that far. So this graph is actually kind of useful for understanding. Blue line is the annual incidence of HIV. Red line is the prevalence, because people with HIV at some point, they started living normal life. So the number, because new people get infected, number of people living with HIV increases. And the last bit before going to the break, I couldn't find a better map, better illustration, but whatever. So first, we're going to talk about sporadic infections. Sporadic infections have no geographical prevalence. Okay? Really the perfect example would be tetanus. Well, we don't have tetanus, but like botulism. Okay? You can get botulism in New Jersey or in California or in Texas with the same probability. Then we have endemic. This is something that happens in a specific geographical area. For instance, coccidioidomycosis, the fungal infection, is endemic to the Southern California. Okay? Or Rocky Mountain spotted fever is endemic for Tennessee and Kentucky in particular. Right? Then we have epidemic. Epidemic is when the number of cases significantly exceeds what you expect. So when you look at the graph on the previous slide here, 2007-2008 season was epidemic season. It was an epidemic of influenza because we have it every year. That year it was bad. Okay? That's epidemic. Now, if tomorrow the outbreak of 50 cases of hepatitis A is discovered in Manor, that's epidemic outbreak as well because we expect none. Clear? Pandemic only refers to spread worldwide. This is where it becomes weird. Look, influenza, does it affect the entire world? I wouldn't call it a pandemic. So I really like the definition, which is quite nice because it rhymes. Pandemic is the type of outbreak that changes how we work, live, and play. Changes the way we work, live, and play. So SARS-CoV-2, definitely a pandemic. Herpes, definitely not. Okay, that makes sense? Okay, let's take a break.